Hello and welcome to Patho Excellence. Today we are learning approach to HP slides under the microscope. In histopathological slide examination, always follow a systematic approach using the microscope objectives in sequence. So let's see what are the different objectives in the microscope. So here you can see 4x, 10x, 40x, 20x and 100x. This is called scanner. This is low power objective lens, medium power, high power, and this is oil immersion lens. For histopathological slide examination, we require 4x, 10x and 40x. So let's start step-by-step approach to identify and diagnose an HP slides. So first step is low power scanning. We use 4x or 10x for the scanning of slides. Here the first step is to scan the slides systematically. Start from one corner, move in a snake-like fashion, look at tissue type, architecture and overall pattern. Second step is identify the organ. Based on the architecture or like lobules, ducts, villi, follicles, glomeruli, you must appreciate what is the organ, because if you know the organ, you can easily reach to the diagnosis. Like suppose you appreciate this is the slide of liver, then you know mostly diagnoses are fatty liver, cirrhosis or hepatocellular carcinoma or hepatitis. So, by identifying the organ, you can narrow down the list of diagnoses. Or you note any special structure, whether capsule is there or not, mucosa, glands, cartilage or bone. Now third step is look for lesion distribution, whether lesion is there which is localized or diffuse, whether focal, multifocal or widespread lesion, is normal architecture preserved or distorted. So we have seen slide thoroughly in scanner, identified the organ. Now we have identified the lesion, and usually we use hematoxylin and eosin stain, which gives cytoplasm pink color and nuclei a purple blue color. Now second step, which is called pattern recognition. So we use medium power, either 10x or 20x, to recognize histological pattern, whether this is inflammatory, neoplastic or degenerative or necrotic, cystic or fibrotic. For tumors, whether it is benign or malignant, which is identifiable by architectural views. Second step, check interface between normal and abnormal tissue. Means sharp border between normal and lesion part. So it is benign. Infiltrative margins means tumor cells are infiltrating in the normal tissue. So it is malignant. For inflammatory lesions, identify the cells like neutrophil, it is acute, lymphocytes, plasma cells, macrophages, it is chronic, granulomatous, it is epithelioid, giant cells if there, it is granulomatous. Then high power view, which is 40x. It gives us better cellular and nuclear details. Nuclear features include size, shape, chromatin pattern, nucleoli, atypia, hyperchromasia, mitotic figures, whether they are normal or atypical mitotic figures. Next step is to identify cytoplasm. What is the amount, color, inclusions or vacuolization? Some special cells, if present, like Reedstone, it gives a clue to Hodgkin's lymphoma. Langerhans giant cell, it is in tuberculosis, malignant cells or mesothelial cells. Background is important. You must write down whether it is necrotic, fibrinous, mucinous or presence of amyloid and inflammatory exudate. Fourth step, once you have examined the slides, you correlate with the clinical information if provided. Like in exam, they may give us age, sex, symptoms or gross picture, then integrate all this data. For example, middle-aged female, thyroid swelling, and if you appreciate papillary structure and often any nuclei, then the diagnosis is definitely papillary thyroid carcinoma. Fifth step is make a differential diagnosis. List 2 to 3 possibilities if not 100% sure. Then you narrow down your diagnosis by architecture, like whether it is lobular, glandular, papillary or in sheets, cell type, epithelial type, mesenchymal, hematopoietic or neural. Facial features like presence of calcification, keratin pulse or type of necrosis. Then final diagnosis. In final diagnosis, you state like what is the organ, what is the pathology and subtype. For example, colon having adenocarcinoma and moderately differentiated, it is a subtype. Most important exam tip, always describe before naming. Means in practical exams, marks come for description, not for just spotting. So you write down what you have seen in low power view, like architecture, what is the organ, lesion distribution. In high power view, cytological details, and finally the diagnosis, which must be concise and complete. This is a quick mnemonic for microscopic exam, like OPD means O for organ identification, then architectural identification, then pathology type, C for cell details and D for diagnosis. This is the practical exam checklist. Already we have discussed the steps. First is the identify the organ, then describe the architecture, then type of pathology, high power view of cellular and nuclear details, correlate with data if provided, and give your final diagnosis. This is the example how you see the slide under scanner view. So in scanner view, this is the part of normal intestine. Here you can appreciate pink muscles and some abnormal areas of dark blue colors in the nesting pattern. So these are the varying size of nest in the muscle area. And if you see in high power view, you can easily appreciate nuclear details. So here it's showing nest of small blue round cell tumors. The cells are round, dark blue in color, and chromatin is salt and paper like. So this is the new benign tumor of intestine. This is again a deeper view of showing here. This is the normal mucosa. This is the submucosa part, and a large area of blue cells in nesting pattern, in trabecular pattern, you can appreciate. This is the infiltrative pattern, infiltrating whole thickness of intestine is involved by this large tumor. And this is the overview. Here we can appreciate tumor part having glandular pattern, nesting pattern, some trabecular pattern, and blue round tumor cells having salt and pepper chromatin. This is how you can make a list of lesions or slide, and what will you see in scanner view, in low to medium power view and high power view for the diagnosis. Like these are some lesions: acute inflammation, granuloma, fatty change, amyloid gangrene, and these are the different appearance of different lesion in all three objective lenses. Same thing in systemic pathology. Some lesions of lung like lobar pneumonia and tuberculosis, different features in these all different objectives. We can appreciate when we see microscopy in the same approach, then we can easily diagnose the lesion.